Hello, I'm David Spiker, and I will be presenting two lectures on proteomics. The first lecture will be an overview and the basics of proteomics. The central dogma is DNA is converted to RNA and is converted to proteins. So we'll be talking primarily about the protein complement of the genome, which is a term that was first coined in the mid-1990s. And indeed, proteomics is a relatively new field that started in the mid-1990s. An important difference between proteomes and genomes is that proteomes are highly dynamic. Genome is pretty static over the lifetime of an organism, but proteomes are constantly changing. The proteome at one moment is different than a proteome a short time later. So we have a nearly infinite number of proteomes. As indicated by the picture, this caterpillar and the monarch butterfly have the same genome, but the proteomes are quite different, and it is not even protein levels that define phenotype, but protein activities, which are not the same as the protein level. So what is proteomics? Proteomics is any large-scale or systematic characterization of a proteome or a well-defined subproteome of a cell, tissue, or simple organism. And we can ask global or targeted questions, and we'll go into more detail in a few minutes as to the difference between these. At one point, proteomics was thought to be a fancy new name for protein biochemistry, but the two are really quite different. Protein biochemistry is based on prior knowledge and usually is a hypothesis testing type of study. In contrast, proteomics is unbiased, it is hypothesis generating, and it is not based upon prior knowledge. An example is what proteins or gene expression of which genes are important in the development of metastatic cancer. A global question attempts to analyze the entire composition or changes in the protein complement of a cell, tissue, or organism. Analyzing the entire proteome is very feasible for prokaryotes and yeast. It is more challenging for higher organisms, but as a result of recent advances in analytical technologies as well as mass spectrometers, which are the key analysis method for most proteome methods, we now can analyze essentially all of the gene products of a mammalian cell line, for example. However, such proteome analyses, where we're globally analyzing the protein complement of a cell line, will not directly give us much information on post-translational modifications or splice variants. When conducting global proteomic studies, invariably we're going to observe differences. The real challenge is to interpret the significance and to understand what are the key factors or the drivers that define the observed biology or disease. There are frequently many changes that are considered passengers that are just along for the ride, so to speak. Now, systems biology types of analyses, such as network and pathway analyses, can help define what are some of the key changes in a proteome. However, follow-up validations studies are still required to test and determine which are the key drivers. Another type of proteomics question is targeted proteomics, and here we're analyzing a particular well-defined subproteome. Some examples include analysis of organelles or analysis of macromolecular complexes or cellular machines. Examples of cellular machines include the proteasome, ribosomes, and the nuclear pore complex. We also can define and isolate specific protein groups. Classical examples are phosphoproteomes, glycoproteomes, the cell surface proteome, as well as functional classes of proteins such as cysteine containing proteases that have cysteine at the active site. Some two key factors when conducting targeted proteomics are to analyze the reproducibility of the isolates as well as distinguishing nonspecific interactions and contaminants from moderate affinity interactions. Why are the above important? If I have variations in the isolation of a subproteome, those variations in the isolation are going to show up as differences in the protein profile. They're artifactual differences rather than biologically related. An advantage of asking targeted proteomics questions is the data sets are simpler and the question is more focused, and therefore it's easier to correlate the changes with biological phenotypes. Proteomics provides some advantages over nucleotide methods, nucleic acid methods, including microarrays and deep sequencing. Protein activities, as mentioned previously, actually define the phenotype, but messenger RNA levels and not even protein levels will necessarily always correlate with the protein activity. In part, this is because most proteins are post-translationally modified, and post-translational modifications usually regulate function. The function of proteins also can be affected by changes in subcellular localization. For these types of questions, proteomics is often the only method of interrogating changes. There are, of course, some disadvantages of protein profiling compared to genomic approaches, and one is that we do not have an equivalent of PCR, and so we cannot amplify proteins. As a result, proteomic analyses are oftentimes less sensitive and more expensive, and will not necessarily easily survey as large a portion of the mammalian omic space. Most methods are minimally automated, and throughput is somewhat lower than many genomic methods. However, the endpoints of the studies usually are very similar. We end up with a list of genes or a list of associated proteins. We then need to correlate these changes with biology or disease, and we have similar challenges. Mining databases for existing knowledge about the identified key proteins or genes, determining what changes are worth following up, determining and distinguishing between drivers and passengers, as previously indicated, and it's necessary to validate the results that we've obtained by an independent method. So what is the scope of the human proteome? The human genome is now pretty well defined. It's around 21,000, 22,000 genes. There are some few remaining uncertainties, but we clearly are in the low 20,000s in terms of numbers of genes. Many genes are alternatively spliced, and therefore the number of transcripts is on the order of about 200,000. In addition, most of these transcripts are post-translationally modified. There are not good estimates as to how many post-translational modifications occur on average per transcript. However, a guess of about 10 or more seems reasonable, and therefore the total number of unique protein species that may affect biology in the case of humans is on the order of several million. In addition to this great complexity, we also have a dynamic range challenge. In a cell line, the copy number of individual proteins might range from approximately 100 up to 10 million or more copies per cell. In biological fluids, the dynamic range of protein concentrations is even greater. Estimates for plasma are 10 to the 10th to 10 to the 12th, and to some extent this is limited by the detection level, and as we become more and more sensitive in terms of detecting low abundance proteins, probably the dynamic range in biological fluids is going to be even wider than 10 to the 12th. Now, how many proteins might we expect per cell type? estimates of how many genes are expressed are 10 to 12,000 genes or more. And not all of these are post-translationally modified to a maximum extent, so perhaps 50 to 100,000 unique protein components may be present in a mammalian cell line. Proteomics capacity has, without fractionation, has a limit of about 4 orders of magnitude and hundreds to maybe up to 1,000 proteins. So clearly, fractionation is necessary to interrogate a large percentage of the human genome. One question we might ask is, why did proteomics develop in the mid-1990s and not sooner? There's a number of reasons. The major ones are listed on this slide, and the really essential components are the availability of complete or nearly complete genomes and mass spectrometry advances. The other factors are also important, but it's really these two advances that are absolutely key to the birth of the field of proteomics. As an illustration, in the mid-1980s, I developed a proteomics type question. One of my colleagues had many different cell lines at every stage of melanoma progression, starting with normal nevi from normal melanocytes to non-cancerous nevi to early, mid, and late stage cancer. We were experts at that time at using two-dimensional gels and thought that we would compare these different cell lines and determine what changes are important as the cells progress through the various stages of melanoma development. We analyzed the cell lines using two-dimensional gels. We saw many spots change. We very laboriously worked at obtaining partial sequencing using the technology of that time, which was Edmund sequencing. We obtained little snippets of protein sequence, and we searched databases. Back in the mid to late 1980s, if you took a human sequence and searched it against the database, there was about a 20% chance that you would actually identify the protein or a close homologue. The majority of proteins, the sequence was not available. At that time, it took a graduate student or a postdoc a year or more to clone an individual gene, and if we identified 50 spots and 40 of them were uncharacterized, we weren't going to spend 40 person-years cloning these genes and characterizing them. So we shelved that project because the technologies were not available. We now have mass spectrometry, which allows us to much more rapidly identify those spots off the gel. And now when we search those sequences against the complete human database, we will unambiguously identify the protein that corresponds to that two-dimensional spot. So as implied by that short story, mass spectrometry is a fundamental proteomics tool. Without it, we couldn't do most proteomic types of analyses. Now, in the early pre-1990s, mass spectrometry was very exotic. It was largely limited to small molecules in a few highly specialized laboratories around the world, and proteins and peptides just could not feasibly be analyzed. In the 1990s, there was a revolution which was fostered by development of two soft ionization techniques, MALDI and ESI. MALDI stands for Matrix-Assisted Laser Desorption Ionization, and ESI stands for Electrospray Interface. These two discoveries were so important and fundamental that a few years later, the Chemistry Nobel Prize was awarded to leading developers of these techniques. This technology and commercial instruments providing this technology have rapidly evolved since the 1990s, particularly for electrospray mass spectrometry. Now, ultimately, a mass spectrometer is simply a very expensive and rather large bathroom scale. And actually, all you get is masses, or more precisely, M over C, mass over charge. You don't even get the mass, but you usually can infer or determine what the charge is, and so you indirectly determine the mass. So this is a single physical parameter, but it's actually a very powerful technique in the context of experimental design and with the availability of complete sequence databases for organisms of interest. Just briefly, instrument design can be divided into three components. I need a method of getting my ions into the mass spectrometer, and this is ESI and MALDI as previously discussed. I then need to separate my ions or have a method of determining the mass over charge, and then I need to detect the ions. Ion detectors are pretty vanilla. They simply tell you, well, do I have an ion here or don't I under this circumstance? There's several different ways to separate ions. You can do a separation based on time of flight, or various mechanisms by which ions are manipulated in magnetic fields. Initially, instruments came in two varieties. MALDI, associated with time of flight, or electrospray coupled with magnetic fields, usually quadrupoles or octopoles. However, now there's a lot of mixing and matching going on, and one can obtain either interface with virtually any type of separation mode, and some instruments are available with both interfaces on the same instrument. There are four key factors in mass spectrometry analysis, and this is a case where more is always better, and you can't have too much of a good thing. First, resolution. This is can I resolve monoisotopic masses or not? And we'll go into more detail as to what monoisotopic masses are. Mass accuracy. Mass accuracy can be expressed in parts per million or as a percent, but more commonly with the advent of highly accurate mass spectrometers over the last 5 to 10 years, most of the time we refer to mass accuracy in the parts per million category. Sensitivity started out in the picomole level, is now down frequently to the attomole level, and some heroic efforts are done are performed at even higher sensitivity. Another factor is duty cycle. How fast can I acquire data? Proteomes are incredibly complex in most cases, and therefore the speed with which I can acquire data is critically important. I'd like to briefly talk about the two interfaces that we have available. One is MALDI time of flight, or first of all, MALDI, and regardless of the separation method. MALDI is simple, easy, and fast. It is also relatively tolerant of salts and buffers, although large amounts of salts and some buffers and some other components that might be in your sample can greatly suppress signals, so it's not completely tolerant of the ionic environment. It consumes a very small amount of sample, half a microliter to a microliter of a protein or a peptide solution is frequently enough, and sometimes these samples need to be diluted. And one can achieve isotopic resolution below 5 to 15,000 depending upon the instrument. It is actually your only mass spectrometry option for large proteins, although ESI of larger proteins can now be achieved with special techniques and with some of the most current instruments. There are some disadvantages to MALDI. One is that the optimal amount for analysis is quite variable. You never know what the optimal amount is, so you have to experimentally test it. Some ions can be selectively suppressed, so you're never going to see everything that's there. And it has moderate accuracy compared with the best ESI methods. It also has poor resolution at higher masses, primarily because in MALDI, proteins and peptides have primarily a single charge associated with them. ESI is particularly interesting and increasingly powerful because it continues to rapidly improve. There have been major advances in ESI instruments from the mid-1990s to the present. to the point where every 1 to 3 years, a major technological advance in the instrumentation area has occurred, and these improvements often result in order of magnitude level improvements in either sensitivity or duty cycle. ESI instruments are usually interfaced with nanocapillary HPLCs, and HPLC using reverse phase is a very robust way of separating peptides. To maximize sensitivity, the current standard is to use 75 micron diameter reverse phase columns, C18 or similar types of columns, with very small particle sizes. These small particle sizes generate high pressures, and so now the standard is to use nano UPLCs instead of HPLCs. A UPLC stands for ultra high pressure liquid chromatography. Current instruments are very fast, very sensitive, and very accurate, and have very high resolution. So they have all 4 of those key factors that we talked about earlier. Some of the state-of-the-art instruments include hybrid linear ion traps, such as the Orbitrap Elite and other members of the Orbitrap family, as well as ESI QTOF type instruments. It has relatively few disadvantages. One is that operating ESI MS on a hybrid instrument is more complex and with many more parameters, and therefore it is much more difficult to operate a ESI instrument than a multi-TOF instrument. Also, the instruments are very expensive, and because the technologies are rapidly evolving, it's a real challenge for a proteomics laboratory to consistently obtain the newest technologies. This diagram represents the basic schematic of a LC MS system. We have a nanocapillary HPLC, or more likely a UPLC, which is operating at a in the nanoliter per mil range. It is interfaced with a refrigerated auto sampler, which stores your samples so that you can operate 24/7, and typically a few microliters, 1 to 10 microliters of sample are injected onto a trap column. This trap column will store this large volume sample because the volume being injected exceeds the volume of the analytical column. After trapping the sample on the trap column at a relatively high flow rate, that is perhaps 1 microliter per minute, a valve is switched, and now the nanoliter per minute flow rate washes or elutes the peptides or protein from the trap column onto the analytical column. The end of the analytical column is drawn into a fine point, and a fine spray is emitted. This spray contains ions as well as non-ionic components. There is a moderate voltage differential between the column and the mass spectrometer, and so we have an electrical field of perhaps 1,000 volts, give or take a bit, and depending upon the polarity, if we have a negative electrode here, all the positive ions will head towards the mass spectrometer. These positive ions are desolvated as a result of being sprayed into the atmosphere, as well as they enter the vacuum of the mass spectrometer, and we have naked positive ions that enter the gas phase, that are in the gas phase at this point in the vacuum of the mass spectrometer. We briefly talked about resolution and monoisotopic masses. What do we mean by this? Proteins and peptides have isotopic envelopes that are caused by the fact that the atoms that comprise proteins, such as carbon, hydrogen, oxygen, nitrogen, and sulfur, exist in multiple isotopic forms. There is a monoisotopic major component, and additional minor forms which are somewhat heavier because they have one or several additional neutrons. Of these atoms that are the major components of proteins, that are the components of the backbone of proteins, most of this envelope, this isotopic envelope, is contributed by C13, which has a natural abundance of 1.1%. And so, if we have high resolution, as indicated by the very sharp peaks, we will see 1, 2, 3, 4, 5 peaks. These 5 peaks are the monoisotopic peak, a peak that is 1 neutron heavier, 2 neutrons heavier, and so forth. We frequently refer to these, since C13 is the most common, as the monoisotopic peak, the 1C13, 2C13, and so forth. However, recognizing that this peak that these peaks could be contributed by the heavier isotopes of other atoms that constitute the protein. As resolution decreases, our ability to resolve this envelope decreases. And so at high resolutions are 60,000, we have the tightest peaks. At a resolution of 6,000, we have the broader peaks indicated here. And if we have a resolution of 1,000, we actually do not resolve any of the isotopic peak. Instead, we have this continuous pattern. Having a little problem with the cursor here. Sorry about that, folks. So we have this very low resolution scan where we don't see the isotopic envelope, and we would then estimate the centroid and obtain an average mass. Determining where the centroid of this asymmetric peak is performed with moderate precision, and therefore the average mass is always less accurate than the monoisotopic mass. Another important point here is as masses increase, or mass over charge increases, you'll notice that the monoisotopic peak gets smaller because the probability I'll have at least one heavy atom in the peptide decreases. These are some actual data which were acquired under high resolution conditions in the lower panels and low resolution conditions in the upper panels. And you can see that we got peaks that are very similar to the idealized version that were shown on the previous slide. This is additional data, and this points out an important point that as my signal intensity goes down and as my masses increase, I reach a point where it is difficult to determine which is the monoisotopic peak. Whereas if I have a strong signal, and particularly if the m over z is the mass of the peptide is small, it's very clear that the first and largest peak is the monoisotopic mass. A couple of basics about mass spectrometry. As mentioned previously, we're always measuring mass over charge. We're not measuring mass directly. And we are getting these biological molecules into the gas phase in a vacuum. We're measuring ions. We can analyze in positive or negative ion mode. However, if a compound is not charged, we will not see it in the mass spectrometer. There are also several levels of mass analysis. One is to analyze the ions that were introduced into the mass spectrometer. So this is MS analysis. Whatever ions are introduced are measured. This could be a peptide or a protein, or a group of peptides or proteins. The next level is MSMS, also referred to as MS2 or tandem mass spectrometry, where we measure the mass of the parent. We fragment that parent ion, a selected parent ion in the mass spectrometer, and measure the masses of the product ions. This can continue to go on to higher levels, at least in theory, and with more modern instruments, MS3 and possibly even MS4 are realistic. But as I go up and continue to fragment the fragments, the signal goes down, and I'll reach a point where I can no longer detect. So those are the basics of mass spectrometry. How do we identify proteins, and how does this relate to proteomics? There are several ways of identifying proteins. An old method, which is introduced here for historical reasons, is mass fingerprinting. We're simply using tryptic peptide masses measured in a multi-time-of-flight instrument were used. This is quick and easy, but it's not very reliable. There are lots of false positives and negatives, and as tandem mass spectrometry or MSMS methods have expanded, the use of this method has largely died out. It is still occasionally used for analysis of two-dimensional gel spots because of its speed and ease of use. However, in large part, this is an old technique that you should typically not consider. MSMS is the preferred method to analyze peptides, and it uses tryptic peptide masses as well as the fragmentation of those tryptic peptides to identify the peptide relative to a sequence database. Tandem mass spectrometry is a three-step process. I measure precursor ions, the ions that are introduced in the instrument. I select a specific precursor ion of interest. I fragment it in the mass spectrometer. This is called collision-induced dissociation. I'm exciting the molecule by increasing the radio frequency and also introducing an inert gas such as helium and causing a collision between the helium ions and the peptide ions, which results in dissociation of the peptide into fragments. I then measure the masses or the mass over charge of the product ions. This type of measurement can be conducted in tandem in space using multiple mass analyzers, such as a triple quadrupole instrument or an instrument with two time-of-flight sectors. Another is tandem in time using ion traps or other methods of trapping or sequestering and selecting ions. An advantage of tandem mass spectrometry is it produces sequence-related information for peptides and proteins, even in relatively complex mixtures. This results in a high-confidence identification of peptides and subsequently proteins, as well as characterization of post-translational modifications. This process is illustrated in this slide, where a 5-residue peptide is schematically represented at the top. This representation is indicates that there are 3 different places where I could fragment along the backbone of the protein. Ion traps and a number of other common fragmentation techniques fragment at the peptide bond, and this produces a series of ions that are labeled as B ions for those originating at the N-terminus of the peptide, and the complementary Y ions extend from the bond breakage to the C-terminal of the peptide. Hence, I have a series of B ions, as illustrated in the lower part of the slide, and the complementary Y ions, all of which have sequence information because they differ in mass by an amino acid residue mass. This slide illustrates a chromatogram in the upper panel, where now instead of a UV tracing, as is often the case with HPLC, we now are using the mass spectrometer, and the y-axis is relative abundance. We have peaks very reminiscent of peptide peaks in a peptide HPLC map using UV detection. However, if you note where the red cursor is located, it actually is located in a valley where you would expect nothing much to be present, but due to the high sensitivity of the mass spectrometer and the fact this is a very complex sample, that is not the case. And the lower panel shows a full scan, that is an MS scan, of the ions that are emerging at the place indicated by the red cursor in the upper panel. We then can select one of the ions in that panel and fragment it and obtain an MSMS spectra, which is illustrated here, and using software can identify the B and Y ions and the sequence that is represented by that peptide MSMS spectra. The basic steps in protein identification are illustrated. Excise stain proteins from gels or digest the protein or take a protein's sample in solution, digest it with trypsin or another protease. Trypsin is typically used for most proteomic applications where digestion occurs on the C-terminal side of lysine ARG, and we then use nano-LC to separate tryptic peptides, analyze on an MSMS capable instrument, and then do a database search against a appropriate sequence database with an algorithm such as sequest or mascot. These algorithms use both the peptide mass, the precursor mass, as well as the fragment ion mass, which is sequence related. This is not peptide sequencing per se. You're actually correlating sequence related information to sequences as illustrated on this slide. So we've acquired our peptide masses and MSMS spectra, and we do a correlation to theoretical MSMS spectra. Critical parameters to consider are that databases are messy. They're non-redundant. Non-redundant databases are actually redundant. Single protein can be represented by multiple database entries. We have the issue of homologs and splice forms because we're identifying peptides and inferring the proteins that are present by the peptides that are identified. One of the problems is that peptides are shared among homologs and related protein sequence domains. Interestingly, some proteins that we identify by mass spectrometry are actually eliminated from newer database versions, which indicate that the interpretation of inference of protein sequences from available gene sequences is not complete. Search algorithms compare the MSMS patterns. For this reason, we always get the best answer. It's not necessarily the correct one. A critical factor to keep in mind is that if a sequence isn't in the database, it's impossible to get a correct answer for that peptide. IDs are based on the peptide level in most proteomic analyses, and so we need to infer what proteins are present. Many peptides are common to multiple proteins. Inferring which specific protein family are actually present in a sequence, in a sample, will be imperfect. One key question is, what database do I use? Do I use a single species versus all species? An important consideration is that if I use a single species database for a search, I may have contaminants from the environment or sample-related contaminants that are present in my sample, and if I don't consider those sequences in my database search, I'm guaranteed to get a wrong answer because that sequence is not in the database, and the algorithm will give me the best possible answer. Should I use full tryptic specificity, partial tryptic specificity, or no enzyme? There also are incomplete cleavages, although trypsin is one of the more robust proteases available. I can also have modifications, both post-translational modifications as well as artifactual oxidation of sensitive residues such as methionine. The major post-search strategy is to minimize false positives while maximizing true positives. There are a number of different strategies that have been developed to maximize depth of sequence coverage, and this is an active area of ongoing research in proteomics as new and better tools are being constantly developed. I also have a choice of searching different mass accuracies. An advantage of high mass accuracy data, particularly for my precursor ion, is my search space is greatly diminished because the possibilities of peptides in the database that are going to fit an observed mass within a certain mass error obviously decrease as the mass error becomes smaller. Mass accuracy is extremely useful in filtering data and increasing the confidence of an identified peptide. If I obtain a mass analysis with a mass error of about 5 parts per million or less, and I identify a peptide by a sequence search, and the mass error is 14 parts per million, this is almost certainly an incorrect identification. Either that or my mass accuracy is not as good as I think it is. Post-search data filtering is an important area of research, and it will greatly influence the results that one gets. If I use low stringency analysis, I will identify more proteins, more peptides and more proteins, but I will have a lot of junk in that data set because a large number of those identifications will be incorrect. A common current practice is to estimate false positive rates from a decoy database, which is typically the same database where all the proteins are. Sequences have been reversed, and that reverse database is appended to the forward database. I then can calculate a false discovery rate based upon how many identifications occur to the decoy database. False discovery rates in the order of 1% or less are typically the norm in most current proteomic analyses. In addition, we typically report proteins identified by 2 or more peptides because one-hit proteins or one-hit wonders are frequently wrong. Some points to remember are that ESI-MS is more commonly used than MALDI-MS, especially for peptide mixtures or small proteins. MALDI-MS is easy, quick screen for purified peptides and proteins, and used to be the only choice for larger proteins, although the ability of ESI instruments to analyze larger proteins is continually evolving. A key point to keep in mind is you never see everything. Protein coverage usually maxes out at 30 to 80% of the protein sequence. When using a single protease, one can expand this frequently by using 2 different proteases in separate digests and separate MS/MS analyses. Up to a point, the more sample you have, the more you will see, and if it's a complex sample, the more you fractionate, the more you will see. Maximizing mass accuracy, resolution, sensitivity, and speed are important parameters. Speed is particularly important for very complex